Hey everyone, Fiona's back with another presentation. And again, like I said, I wanted to upload a couple videos of myself presenting some research so that if there are any middle schoolers or high schoolers out there who missed their science fair and don't get to present their work as a result of this whole coronavirus issue, then maybe they'll feel more comfortable if they see an example of it. And again, you don't have to post a video. You can upload an abstract. You can write a little description, maybe include some pictures. And me and my friends would be happy to comment positive things, hype you up, as well as maybe comment with some questions. And you can field those questions and we can all communicate some science. Okay, anyway. This time I'm going to be talking about a bit of research I did, again as a graduate student, but in collaboration with a group at the University of South Florida that's in natural products chemistry, which is a really fascinating area of chemistry. And I'm going to try to go faster this time because last time I took forever. But anyway, okay, so quick rundown about natural products chemistry. Natural products chemistry is a subdiscipline in chemistry that aims to isolate and identify new chemistry from the environment. So they go out in different areas of the environment, different parts of the environment that might have a lot of ecological diversity, and they find species, be it invertebrate species, vertebrate species, whatever, and then they isolate metabolites, which are just chemistries that those biological species produce. They isolate those and characterize them. Characterize meaning solve the structure of them. So, one such group is a group that we collaborate with at the University of South Florida, and that's the group of Bill J. Baker at the University of South Florida. And their group is really cool because what they do is they go to Antarctica. And Antarctica is really fascinating for chemistry because a lot of the animals and such that live there are sedentary invertebrates. So invertebrates like sponges, corals, maybe not corals, it's too cold. But, anyway, invertebrates that live on the seafloor and they have to defend themselves by producing chemistry that fend off, predators and whatnot. Right? So they produce molecules that fend off predators, which is really, really cool. So one such molecule that they isolated was this molecule here called arabucinone. I'm trying to get my laser back over there. Arabucinone. And arabucinone is really fascinating. It was isolated from a sea sponge in Antarctica. Okay? And what they found after they isolated it is that if you provided arabucinone to crabs to eat, for example, like if you fed crabs a diet including arabucinone, the crabs would stop molting, right? The molting, the process of shedding their shell, and then they died because without molting they weren't able to develop and they grew into their shell and died. So this is really fascinating. Sad for the crabs, but cool for us because crabs are closely related to insects like pest insects, right? So if this molecule could potentially be somehow used as like a pesticide, right? That doesn't affect humans, doesn't affect mammals, but affects pest species, then we could use it to safely spray on crops. Right? And keep pests away from crops, but feel a little bit more confident that we're not affecting our own biology by introducing this into our diet. Okay, great. So, now, that was a bit of an experimental backdrop for then what comes. Again, I'm a computational chemist, so I don't work with isolates of arabucinone, I don't work with crabs, I don't work with sponges, but what I do is I can model proteins. So this pathway is the main pathway responsible for molting in crustaceans. Now this might look a little bit like, Fiona, this doesn't make any sense to me. These are steroidal compounds found in crustaceans. And what do we know about steroids? They're responsible for development in all walks of life. They are responsible, like humans have estrogen, testosterone, progesterone, right? These are compounds related to stuff that looks like this. And these things trigger developmental processes in animals, okay? So when this compound gets synthesized in crabs, it then binds to an enzyme, this enzyme here, CYP315A1, that's just the name of this enzyme. This enzyme then turns it into this compound, ecdysone. This, and then ecdysone then binds to this protein, CYP314A1, and 314 turns it into this compound, and this compound is the compound that stimulates molting in crustaceans. So, if arabucinone is somehow inhibiting molting in crustaceans, then it must be somehow inhibiting this pathway. And it's likely inhibiting, therefore, the production of this molecule called 20-hydroxyecdysone. So, from a computational standpoint, we wanted to conduct docking studies to understand how, trying to get the cursor again, understand how this molecule could bind to these proteins and therefore inhibit them, right? And furthermore, which does it bind to? Is it more likely to bind to one over the other? And again, we want to understand this process because if we understand this process, we can possibly improve the binding of arabucinone to one of these compounds, or one of these proteins, and therefore improve its efficacy as a potential pesticide, right? So this is all really cool stuff. Okay, great. Now, and again, this goes back to what I was saying before. If you look at how my poster's, while cluttered, my poster kind of looks like what you guys make for your fairs, right? Got an abstract, introduction, methods, and then I start with the results. Okay, so I'm going to skip to the docking results here. Now, another key component of this work was in constructing homology models. Okay, so that's what these things are up here. The thing is, there are no known crystal structures of these proteins, 315 and 314A1. Crystal structures are pictures of the protein identified experimentally. There are no known crystal structures of these proteins for crustaceans, so what we did was we constructed computer models based on the one-dimensional amino acid sequence. We predicted the structure. Okay, I'll include links to all this, these topics. Okay. And then now what we did was we conducted, just trying to figure out where the results are that I want to show. Okay, yeah. We conducted docking studies where we docked, in this case, we docked arabucinone into CYP315A1, that's this binding site here, and we docked arabucinone into CYP314A1. And what's key is, again, this predicted binding free energy here, okay? Now, these are on a different scale than they were in the PETACE example, so just because it says -58 here and the PETACE example said -8, that's not really something we should pay attention to. What we should instead pay attention to is the relative difference between this -58 and this -53. And remember what I said is that everything likes to tend, in nature, things tend toward the lowest energy. So what that suggests is that the interaction between arabucinone in CYP315A1 is more favorable than the interaction of arabucinone in CYP314A1. And furthermore, what we hypothesize is key is once again an aromatic interaction. Do you guys remember we had aromatic interactions in the PETACE and METACE example where we had those amino acids in the binding site that were forming aromatic interactions. We have that same case here, okay? This is a phenylalanine amino acid in the protein. And arabucinone is forming what's called a edge-to-face aromatic interaction. And those can contribute, about 2 to 3 kcals per mole, okay, in the binding for energy, okay? And however, over here in the 314 example, there's no such additional aromatic interaction. So there's aromatic interactions between the ligand, or the small molecule, arabucinone, and this heme cofactor, right? It's sitting on top of the heme cofactor. I'm trying to get the laser here. but there isn't an additional one like there is in 314 or 315. I'm sorry, we have the heme cofactor and an aromatic interaction to this phenylalanine. Okay. Now, if we look at how does the enzyme function normally, so this is the enzyme, and if you look at the yellow atoms, I think, yeah, okay. If you look at the yellow steroidal compound, those yellow atoms, you see that it's docked in a way that it interacts really favorably with CYP3, with this phenylalanine and CYP315A1, meaning that this phenylalanine amino acid is key for the function of this enzyme. And what likely happens is arabicinone binds to this enzyme and interacts with that phenylalanine, preventing the binding of the substrate of the enzyme. So if we go back to the question that we had, which was how does arabicinone inhibit one of these two proteins to inhibit molting, we think what's key is it binds to 315A1 and it interacts with an amino acid in that binding site. And we kind of arrived at that conclusion through docking studies. And there's a lot more data on this poster, but I think I'll just keep it at that because I'm already at over 10 minutes again. Oh boy. Okay, but yeah, so that's another bit of research I've done in my graduate work. And again, if no one else posts videos, at least I get some practice because I have my dissertation defense in 2 weeks and this will be on it. So at least I'm getting some free practice. Okay, team. I'll post another video soon, or at least I'll take another video soon and then I'll probably post them all at the same time. But whatever. Okay. That's that.